All right. Hopefully everybody can hear me. Good afternoon, everybody, here in the room, but also online. My name is Lars Gorsa-Wortmann. I'm the Division Head of Peds Cardiology. And welcome to KCVI Grand Rounds, sponsored this time by Peds Cardiology. Today is also our Kasdan Day, which is a very special day in our division, where we invite an eminent speaker who is really a world expert in the field of Peds cardiology or congenital heart disease or cardiac critical care. And tonight, this afternoon, it's my pleasure to introduce Dr. Martin Tristani-Firouzi. In this role, he really deserves to be mentioned in the lineup of these top experts from around the world that we've been able to recruit for this visiting professorship over the last 5 decades. Dr. Tristani-Firouzi is a professor of pediatrics and pediatric cardiology at the University of Utah, where he is also the co-director of the Center for Genomic Medicine, which is the University of Utah's premier center and really one of the nation's premier centers for genomic innovation discovery and the translation of genomic medicine into improved clinical care and outcomes. His interests are as such, meaning in genomic medicine and the integration into clinical care. He leverages large clinical and population-based databases and explainable artificial intelligence to advance personalized care in pediatric cardiology. He is a very affluent researcher with over 25 years of continued extramural funding. One of those funds includes being a PI on an NHLBI Pediatric Cardiovascular Genetics Consortium project called Bridging the Gap Between Genomics and Clinical Outcomes in Congenital Heart Disease. Dr. Tristani-Firouzi is the PI on multiple ROIs, and has published well over 120 scientific papers. Clinically, he is an electrophysiology expert, and that is also the realm of his talk this afternoon. We heard from him this morning about difficult cases in electrophysiology together with our fellows. And then at the noontime lecture, he enlightened us about artificial intelligence. And tonight, the title of his talk is Human Migration and the course, I'm butchering this up, but the course of alleles that predispose to atrial fibrillation. So with that, I hand it over to Dr. Tristani-Firouzi for his talk.
Great, thank you very much. And thank you members who are in the audience there and individuals who are online. I'm going to start sharing my screen here again. Okay. All right, and then I'll just start the talk here and grab a pointer. There we go, great. All right, so the talk that I'd like to give this afternoon is called Human Migration and Ancestral Origins of an Atrial Fibrillation Risk Allele. I have no relevant disclosures. And I'm going to talk about a paper that we recently published in Nature Communications describing the history and geographical distribution of an atrial fibrillation risk allele in the gene case in Q1. And then in the second half, talk about leveraging ancient genomes, ancient haplotypes, to refine ancestry-specific atrial fibrillation risk. So this first paper was a collaboration between Intermountain Healthcare, the adult cardiologists at Intermountain Healthcare, of course, the University of Utah, and really importantly, Ancestry DNA. And it was this connection with Ancestry DNA, which people may or may not know is a Utah company, that I think really elevated this paper way outside of what we could have, accomplished just with the data that we had here in Utah. And I'll explain what I mean by that. So I know there are all cardiologists in the audience here, and so people don't need to see an ECG necessarily, but for any trainees or medical students that might be in the audience, atrial fibrillation is the most common sustained arrhythmia seen in clinical practice, primarily in the adult cardiology clinics. And AF patients have increased risk of stroke and early cardiovascular death. Patients that have young onset atrial fibrillation have a strong genetic component. And when we think about the genomic architecture of any kind of a disease, there's always, or in many cases, there's a combination of common and rare variants that could contribute to the risk of the disease, in this case, atrial fibrillation. And what this is showing here is the relationship between the allele frequency, meaning more and more common frequency of that particular allele, and the impact of that allele on a particular phenotype. So when we get out here, 70% of the population would carry this genetic variant, and by definition, it is a common variant, and it would have to have low impact, or you'd have too many people in the world with terrible disease. As we move into the less and less common alleles, we get into this rare, very rare space where these rare variants can potentially have a very high impact on a disease space. You can have rare variants which are inconsequential, and you can have rare variants that cause disease. So just because a variant is rare doesn't mean it's damaging, but of damaging variants that are rare, they always have a very high impact. And this would be the space of long QT syndrome, CPVT that we talked about this morning, and other inherited disorders. So these common variants out here, where you have 20 to 70% of the population, these variants are used in what are called genome-wide association studies. And this is a GWAS study looking at the risk of atrial fibrillation. And when you have these common variants, you need lots and lots of people. So look at this, 120,000 individuals with atrial fibrillation. These are almost all of European descent. And what these GWAS studies do is they say, okay, well, of all these genetic variants that are present in a SNP array, which are the ones that are statistically associated with the phenotype of interest, in this case, atrial fibrillation? And what we have listed in this plot, this is called a Manhattan plot, we have individual genetic variants, which are the little dots. This is chromosome 1 through chromosome 22. And then we have a line drawn here, which is called the genome-wide significance, because we need to correct for multiple tests. So if you're doing 20,000 tests, 20,000 different genes, you need to correct for that, or you're going to have, falsely inflated values. So then on this axis, what we have is the negative log 10 p-value. So this is 10 to the minus 15. This is 10 to the minus 50. This is 10 to the minus 55. And this is the negative log 10 p-value for the statistical association. And what you can see is there's a whole number of genetic variants here across a bunch of different genes. One stands out very, very prominently. This is PITX2. This is a transcription factor which is really important for early heart formation, especially the left atrium and especially the pulmonary veins. And so this is why the PITX2 signal is so strongly statistically associated with atrial fibrillation. Now, there's even bigger data sets, and this association is like 10 to the minus 300. Now, just because it has a very strong statistical association doesn't mean that has a very strong impact. So if you're a carrier of this PITX variant, your risk of atrial fibrillation is only twofold greater than the general population. But that's the way these common genetic variants work. They cannot have large impact. So this is a study looking at, again, GWAS, the common variants. Now in Utah, we take a different approach, and that is the study of rare disease. And we're especially well suited to study rare disease in Utah because of the family databases and of the individuals that first moved to Utah in the 1800s along what was called the Mormon Pioneer Trail. And these are individuals of Northern European descent, and many of them carried disease genes with them, such as long QT syndrome, cardiomyopathy, breast cancer, colon cancer, et cetera, et cetera. And all of those genes were discovered here in Utah for the reasons of being able to put together these enormous pedigrees. And the way this worked is we followed like this cluster of patients, this cluster of patients, and this cluster of patients separately, and they all had this phenotype of young onset atrial fibrillation. And then what we did is we queried a database called the Utah Population Database, which is the largest genealogical database in the world linked to medical records. And this database said, hey, these three clusters are all related to a common ancestor who had date of birth in the early 1800s. And so you can see for these early individuals, there's no medical information available. And as we come down this pedigree, we start to see some AF history. We don't know anything about it, whether it was young onset or not. But as we get lower and lower in the pedigrees, meaning more and more current, we find that people who have this red box here had atrial fibrillation younger than 60. And in many cases, it was actually atrial fibrillation in their 20s. So everyone, not everyone, but a very large number of the population will get atrial fibrillation when you're 70 and 80 years of age, but almost no one should get atrial fibrillation in their 20s. That's a genetic disorder. So we put together this large pedigree here, and then we were able to assay like what would be the gene that's associated and causing that atrial fibrillation phenotype. And it turned out that casein Q1, which is an ion channel in the heart, part of the IKS channel, was the causative gene, and the mutation resided in the casein Q1 gene. Now, mutations in casein Q1 also cause long QT syndrome. That is when the mutation causes a loss of function. When the mutation causes a gain of function, then you get short QT syndrome or young onset atrial fibrillation, and that's the case here. So what we're looking at over here is slight different variation in the pedigree. So it's basically the same data that we see here, but this includes all the unaffected people as well. And so here's that central, the Mormon pioneers here, and then this is the trajectory of the individuals that carry that variant and the disease into, the 21st century, basically. And of these two individuals, there's almost 3,000 offspring. But what you can see is that, when you think about sort of preventive measures and who to screen, we really only need to screen people in these, specific families, because all these other individuals, they don't carry the atrial fibrillation risk allele. So the next thing we did is we used the IPS technology in order to generate stem cells, human-induced pluripotent stem cells from individuals in this family that carry the mutation. And basically what we do is we do the standard things. We infect the patient's blood cells with a virus that contains these 4 factors. They're called the Yamanaka factors. And Yamanaka was one of the 2 people that got the Nobel Prize in medicine and chemistry for creating the ability to generate these inducible stem cells. So we target the peripheral blood mononuclear cells, and when they're transduced with this virus here, then you get these iPS colonies that we grow up, and then we differentiate into beating heart cells. So this is what those beating heart cells look like, and it looks like I can't activate this because I've got a laser printer or laser pointer there. But basically what you would see if this movie played correctly would be waves of beating cells under the microscope. And it's the most incredible thing to see when you see these cells beating and realize that these are from a specific patient and they're beating in these waves. So what we found when we disperse these cells into single cells and then measure the action potentials of the individual cells is for the control patients, we generated, these are in red, all the individual atrial action potentials for this individual. This is another healthy control individual that doesn't carry the mutation in the family. And then the black is the mean of all those tracings. And we compare it to 2 patients that are heterozygous carriers of the mutation, which is an arginine to histidine at position 231 in the casein Q1 channel. And you can see that these atrial-like cells have shorter action potentials. And we look at the mean data, we see indeed that these patients had shorter action potentials than the controls that did not carry that mutation from the same family. So short action potential duration provides the substrate for atrial fibrillation, and let me explain why that is. So this is a CT scan reconstruction of the left atrium. Here are the left-sided pulmonary veins. And if you get a PAC or a focus here that fires, this is going to basically traverse the atrium. And if this depolarizing wave reaches this site right here, but if the site is refractory, then nothing happens. All you get is a PAC. But if you have a very short action potential, that means that this region here is going to have already repolarized fully. So by the time this depolarizing influence gets here, it's ready to depolarize again, and boom, you're off to the races and you get atrial fibrillation. And so that's really the genesis. That's the idea of why this family gets atrial fibrillation. It's because this mutation alters the properties of the channel, of the KCNQ1 channel, resulting in short action potentials, and that lays the substrate for atrial fibrillation. It's a substrate only. You've got to have a PAC or some sort of trigger to get the atrial fibrillation to be sustained. So in the next segment here, I'm going to talk about how we brought in Ancestry into this study and why. And this was a paper that Ancestry had published in Nature Communications, a really amazing story. We're not going to go through all this detail here because it's really complicated, but we're going to focus here down here. And basically what Ancestry does, and so does 23andMe, is they say that they find people that are related to each other because they share genetic information. So if you imagine these two individuals here sharing genetic information, you can break down where their genetic information sort of localizes to in the world. This is Cameroon, Congo, this is Togo. This individual has Togo background as well. And this person sort of falls into a cluster of individuals that are related. But unique to Ancestry is that people who sign up to Ancestry really care about their family tree, and they'll create a family tree, and they'll go after records that Ancestry provides, like birth records from the 1800s, or marriage certificates from whatever time period. And people create their own pedigrees, and they can click an option that says, hey, you can use my information for research. So the people that have agreed to this have allowed their general genetic information to be shared with researchers and these pedigrees to be shared. And what you can do is you can say, okay, this is saying that this individual's, great-great-grandfather was born in London. And that can be used to create maps, birth record maps, and you can look at what happens to people over time. And here's the example that they give in their paper. They're looking 6 to 9 generations ago, which is the 1600s, and showing here is the birth records for individuals who were born in this time period. And these are relatives of people that are in the Ancestry database. And you can see this high concentration of individuals in France. And you can also see that around that era, you already have these individuals going to Quebec, Nova Scotia, and Louisiana. And these are the French Canadians and the Cajuns. And then when we look at different time periods, you're seeing that, nobody is being born in France anymore. They're all being born here in the United States. And so when I saw this paper, I was like, we have to do this with our patient cohort here. And that is the atrial fibrillation family. And so together with Ancestry's help, we basically found relatives of individuals that are at risk for carrying this same atrial fibrillation risk allele, and where were these individuals born, in the 1700s. And what you can see here is Northern Europe is lighting up. And although it's all blocked off here, the main concentration here is in Denmark. So these individuals, ancestors, way back in the 1700s, were being born in Denmark. And the families who created these pedigrees have the birth records to support that. So that's 1700 to 1800. Now we're looking at 1800 to 1850. And what you can see is there's less people being born, less of those relatives being born in Europe, and more of them being born in the US, but not just anywhere in the US. This is exactly the Mormon Trail. This is the trail that Mormons went from New York to Utah. And you can see that lighting up in this time period. And then when we look at birth records for individuals, and these are the relatives born in the late 1800s, now we see, look at this, this is like Utah lighting up like crazy. And so what this is telling us is that we've identified the mutation that causes atrial fibrillation in this family, and we've identified where this mutation came from, meaning Northern Europe, and we can look at the transcontinental migration of these individuals over time. And this is the first time this has ever been done in a disease. And so that was, I think, one of the most exciting things about this study. But there's more. Ancestry is really good at dating the age of genetic variants, and they have a lot of, like technical people who can use tools to infer when in human history did this genetic variant enter. And without going into the details of how this happened, because it's too time-consuming to explain, basically what we timed this to was about 182 generations ago. And if you consider a generation to be 22 years, 25 years of age, it turns out that the mutation entered the human population about 4,500 years ago. And that maps to the Bronze Age. And so when you think about a disease-causing allele, in general, you don't think about that being present in the human population for very long because patients die and they don't reproduce. But when you look at diseases which are later onset in life, like atrial fibrillation, like breast cancer, these individuals have already married and had offspring before they even manifest their disease. So, these patients in our cohort, they develop atrial fibrillation in their 20s. Well, if you think back 100,000 years or whatever, 1,000 years, people were already having offspring at the age of, 16, 17, 18, 19. And so they've passed this disease allele on to the next generation before they actually get the disorder. So there was something that happened very importantly around this time in the Bronze Age, about 5,000 years ago. And that is there was a massive migration and change in the genetic makeup of Europeans because there was a group of individuals called the Yamnayans who figured out how to make a horse and carriage and wheel, and they migrated across Europe and completely replaced the genetic makeup of the early farmers. So prior to this time, Northern Europe was populated by early farmers that had a unique genetic ancestry. And by the time the Yamnaya came, they basically incorporated their genetic material and mated with women of that era, but none of the males' genetic material from that early ancestry, early farmer ancestry survived. Nobody knows why that is. There's no record of like a massive battle where all the men were killed. But that's what basically the genetic history tells us. And for individuals who are interested in linguistics, this has a very interesting consequence for language. The Yamnayas started here. This is right in the Caucasus between the Black Sea and Caspian Sea. And they moved out westward toward Northern Europe. They also moved down to India. And they all of these languages share a same root, which is called the Proto-Indo-European language. And so the question is, did this mutation happen prior to the Yamnayans moving westward? Probably not, because again, a lot of that ancient farmer ancestry has disappeared from the human population. And so what could have happened is they migrated to Northern Europe, and at some point in what is now Northern Europe, one individual spontaneously developed this mutation and then passed it on sequentially over 4,500 years to the people who we saw in Utah today. So we identified the causative, just in summary, we identified the causative mutation in this large Utah family. We characterized the functional consequences using patient-derived cardiac stem cells. And the important thing is here, what had not been done before is we broadened the scope of disease susceptibility alleles into the context of human migration and ancestral origins. And this idea about human population and ancestral origins really sort of sets the stage for the next part of the talk, which is leveraging ancient genomes or ancient haplotypes to refine answer-specific atrial fibrillation risk. So this has always been a sort of a big unknown within the atrial fibrillation field, this differential risk of atrial fibrillation among Black and white individuals. So Black individuals have a higher prevalence of the clinical risk factors for atrial fibrillation. That is, they're more likely to have diabetes, hypertension, or congestive heart failure. But unusually and remarkably, they have a lower incidence of atrial fibrillation compared to whites. Now, Black individuals who have a greater percentage of European ancestry are at increased risk of atrial fibrillation. So this data is telling us that something about African ancestry is protective, and the more European ancestry a Black individual has, the higher the risk of atrial fibrillation that they have. So what can ancient genomes tell us about this? And I want to sort of walk through what this means, but to do it, we have to understand a term called haplotype and what exactly this means. And so just to review your medical school genetics, a haplotype refers to a unique combination of genetic variants that are near each other on a chromosome. So if you've heard about the SNP arrays, and this is how we do, this is a chromosomal microarray as well, but these SNP arrays are specific areas of human variation across the entire genome. And the SNP array is able to detect these specific areas that are known to be variable in humans. And so the array is specifically targeted for areas of variation. And so the sort of the nucleotide that's present on that SNP is sort of brought together in a block called a haplotype. So we see the TGTCT here. That's what we see right here. Now, usually when you have these SNPs right here, everyone will share these same areas right here. That's almost always true. And this allows you to use a SNP array to impute what this sequence is going to be in an individual. But just for the example here in the haplotype, this individual has an A instead of a T, and so their haplotype looks different. This one also has an A here, but has differences between these others, and so this is a different haplotype. Okay, so we all have these haplotypes. So why does this matter? Well, there's something that's called recombination, which tends to erode haplotypes over time. And remember, we all have two chromosomes, right? Two copies of each chromosome, and one from each parent. And at the time of cell division, these chromosomes replicate. And once they replicate, they actually exchange chunks of DNA across each other. So they line up and they exchange these chunks of DNA in what's called recombination. So we're starting off with, say, something that looked like this. They're more homogeneous. And now you can see there's mixtures across here. And then these will separate, and each gamete gets a different rearrangement of genetic material, and that goes into the sperm or the ovum, and that's what gives us, all of our diversity, et cetera, et cetera. But over time, these haplotype blocks get eroded by recombination. But the key thing is that if a gene is located really close together, it will always recombinate together. And that's what's called linkage disequilibrium. And we can use this linkage disequilibrium to understand these blocks of haplotypes. And what we're going to do in these next experiments is we're going to compare the human haplotypes to ancient hominids, meaning Neanderthal haplotypes and Denisovan haplotypes. And the question we're asking is, for this difference between black and whites, remember, it's the individuals that migrated out of Africa that went on to become those of Northern European and Asian descent. And so in this out of African migration, individuals who are not from Africa all contain 1 to 2% of their genome is Neanderthal because of matings that happened, 40,000 years ago. But the Africans don't have that DNA. And so the question is, are there some haplotypes that Northern Europeans inherited from Neanderthals that place them at increased risk of disease compared to Africans? Okay, so the question here is, did Neanderthals or Denisovans, which is another early hominid, did they introduce atrial fibrillation susceptibility loci into humans? And can we use these patterns of linkage disequilibrium to refine and identify these ancestry-specific GWAS hits? So we're looking at the GWAS hits. There's now probably 50 different GWAS hits, and we've looked at all of them for linkage disequilibrium. And this is one of my colleagues who designed a tool that allows us to do this at scale across all the ancient genomes. And we tested this tool called GWAS Haplotyper against a GWAS hit that was found to increase the risk of COVID infection. So during the pandemic, this paper came out, and it basically said that there is a specific GWAS hit which increases your risk of COVID-19 infection, and that exact same haplotype is shared with Neanderthals, and that haplotype is not present in individuals of African descent. And so we basically wanted to prove that our tool worked in that context. So we're looking at the same COVID risk allele. And what we're showing here, this is where the SNP, the SNP that's associated with increased COVID is located. Then we have two things. In yellow, we have the Neanderthal variants, and in green, we have the human variants. And yellow is sticking out because green is hiding behind there. But what this is showing is that these genetic variants from the Neanderthal are all in linkage disequilibrium with this locus here at the COVID risk allele, meaning that if you have this COVID risk allele, Neanderthals have that same allele and they share it this far out. And this is 1.0, this is the LV value. This means that it's 100% in linkage disequilibrium. What you can see when we look sort of on the other side of this gene, boom, we see this falling off very quickly. What it means is that this side of the gene, that haplotype has been chopped up both in humans and in Neanderthals. So this has undergone a lot of recombination events that have chopped away at the haplotype. This haplotype has been preserved for some reason. And when we see a preservation of a haplotype like that, usually it has some functional effect and some importance. So I'm going to quickly go to one of the GWAS hits for atrial fibrillation. This is FBXO32. And this gene is a gene that modulates an important channel in the heart. It's called the small conductance potassium channel, and it influences the expression of that channel. And again, we've got the same thing. We've got yellow being Neanderthal, green being human genes. And now what we're comparing is Europeans and Africans. African ancestry. And what you can see is that for individuals that are of European ancestry, if they have this SNP that increases the risk of atrial fibrillation, you can see that there's this big block of linkage disequilibrium that's shared with Neanderthals. And when we look at the Africans, you don't see that haplotype at all. It doesn't exist. The haplotype has been chopped into tiny pieces, okay? And let me just sort of expand a little bit more on this. So this haplotype is present in humans, and it's present in Neanderthals, and it's present in Denisovans, but it's not present in Africans. So sort of the expectation of how this could happen is that humans that migrated out of Africa mated with a Neanderthal that had Neanderthal and Denisovan genetic background that introduced a series of DNA, right? But this haplotype has survived, and this haplotype is associated with this increased risk of atrial fibrillation. And so this is acting like that COVID-19 risk allele in that it's affecting Europeans, but not Africans. And I'm going to give you one example of a situation where there are no ancestry differences comparing those of European and African descent. And in fact, that suggests that there's a very, very ancient haplotype that is shared by these both Europeans and Africans. And I'll show that it's also shared with Neanderthals, and it's even present to some degree in chimpanzees, which means it's a very, very, very old haplotype. Okay, so this is the is a sodium channel gene, and this GWAS hit is it localizes to a 4.4 kb haplotype that's present in all the ancestries that we looked at. So in here, this is showing both Africans and Caucasian ancestries. So here's the SNP, and you can see this area of linkage disequilibrium. When we look at the actual haplotype itself, you can see that this is shared even with the Denisovans, right? Look at this, ATTTTTT. These are all shared across all hominids. And when you look at chimpanzees, you can see that pieces of that are actually present even in a chimpanzee. It's not exactly the same, but what this is telling you and what this is telling us is that this haplotype is coming from very ancient ancestors that were part of the, even before Neanderthals split from humans. So we're talking, over 1 million years in time. And so the idea is, does this interbreeding that happened between these archaic humans, which are the Denisovans and the Neanderthals, in Eurasia, does this explain ancestry-specific risk of human disease? And I think we've shown that it does for certain alleles in atrial fibrillation, and that can explain why Black individuals have a lower risk of atrial fibrillation, even when they have higher clinical contributors like hypertension, diabetes, et cetera. And so, it was really this series of experiments with ancestry that got us thinking about, human migration, and that led us into this idea about sort of comparing the ancient genome. So it's been really an exciting line of research. And with that, I will end the talk, and I'm happy to answer any questions.
Wow. That was definitely a different talk than we've, I think, ever heard. I found it super interesting. I happen to have sort of an interest in paleontology and anthropology, but I'd never heard much of this before. So really fascinating questions. Well, I have so many questions on how you actually do this research, because I understand so little about it even after your excellent explanations. But I'm just interested in learning, how'd you get into this? You explained a little bit. It sounds like it was the opportunity that you had in Utah and your collaboration with the company. And is this just sort of a research hobby for you, or do you think there is actually a clinical application to this in terms of screening families, screening patients? And I realize those are two different questions.
Yeah. So it is to some degree kind of a hobby. I mean, I'm fascinated by ancient history and ancient genomes and the Neanderthals and sort of the human migrations that happened coming out of Africa. This whole idea of this population of individuals called the Yamnayans that swept across Northern Europe and Asia and completely outcompeted the local people genetically. And so there's an author from Harvard. His name is David Reich, and he has a book on human migration, which is geared for the lay public and has a fair amount of science in it too. But it's a really, really cool read for people interested in the space. So that was sort of my motivation for this. This is a hard thing to get funded for. It just doesn't fit in the wheelhouse of many of the study sections. But I think it's answering interesting questions. But the question also then is, well, how far can you go for that, right? So it came out of the idea about what is it about Black individuals who have African, obviously these are African Americans, but they have varying degrees of African ancestry. And those that have more African ancestry are less likely to get atrial fibrillation. And what would be the genetic explanation for that? So, this can answer something in atrial fibrillation. This can probably answer something in the fields of cardiovascular disease in general, coronary artery disease, hypertension, diabetes, right? There's always going to be some genetic variants which increase these diseases, because when you think back historically, very few people live past the age of 40 when you're talking, 100,000 years ago. And so they're not going to get hypertension. They're not going to get atrial fibrillation. Like, they don't get those late onset diseases. And so if you've got risk alleles, they can circulate freely in the population. But the reason they circulate may be that they have some benefit that we don't fully understand. And so they may have that allele because that benefited them living in the environment that they lived in now. But of course, modern humans, we live in a different environment with like 7-Eleven and McDonald's. And, you eat that kind of food and man, you're going to get obese and you're going to get metabolic syndrome and things like that because everything has changed. So, but that was my motivation for this. This is something that I just love to think about, but it's not clear that it could be supported by an R01.
There's no other questions. I'll ask another one. In pediatric cardiology especially, we often, we struggle to do randomized clinical trials. And when we do, oftentimes they're negative. And then somebody says, well, we just didn't enroll enough patients. And somebody else will say, well, we didn't select our patients carefully enough. So in somewhere in the future, do you foresee this helping with patient selection for treatments and trials, much along the lines of what you just laid out for the atrial fibrillation?
Yeah, where I think it's very important, and in fact, the PHN is paying attention to this is the stronger damaging genetic variants and the impact that that has on patient outcomes. And when you think about if you want to have a drug trial for, let's just say, the trial that the PHN ran on using Udenafil in Fontan patients, right? So if that individual is a carrier of a variant which has impact above and beyond what Udenafil can even do, that can skew the results, and you could get results that say, hey, Udenafil doesn't work at all. But if you stratify the patients by whether they have a genetic variant that's damaging or not, you might find that those that don't carry the variant actually have a benefit. But it's when you look at globally at the individuals in the study, it's sort of like that genetic variant has impact on clinical outcomes or response to therapies that could confound your study. And so the PHN now is being careful about when it does its trial design for therapeutics to make sure that some genetic factors are evaluated.
By Dr. Balaji.
Sorry, Marty, this is maybe a stupid question. How do you get genetic material from Neanderthals and Denisovans?
Yeah, so that resulted in one of the Nobel Prizes going to this investigator, Savant Pablo, who is at the Max Planck Institute in Germany. They get it primarily from the petrous bone. So if you have a skull, apparently the petrous bone is, for whatever reason, the most protected space, and they're able to extract DNA. And it's just remarkable how they do this. And a lot of the DNA is highly degraded, so they use some computational tools to sort of impute what should be there. But the sequencing technology is getting better and better, and now there's thousands of ancient genomes that have been sequenced. And that includes both Neanderthals and ancient humans from like 50,000 years ago.
A really interesting talk. Thanks so much. You kind of talked about this already. I just wanted to, it's interesting to me. So it sounds like a lot of these AFib cases happen at about 20. So I would have expected that would have had some impact on the survival of these people living out there. And I'm sure, like you said, most of them didn't live past 40. But I'm just curious, you kind of already mentioned it, but what have you guys postulated what the potential advantage of this gene would have been, or other genes that it might be linked with that did provide that advantage that would overcome the survival disadvantage?
Yep. Yeah, so there's a possibility that there could be a survival advantage in genes that are nearby the KCNQ1 locus. What I think, though, is I think that this had a definite impact on fitness. But again, because they may have already had children before the age of 20, that they could have passed this allele on to the next generation. And then they get into their 20s and 30s and they have atrial fibrillation, they feel terrible, they have a stroke and they die, or they're incredibly debilitated. So I do think that this is an allele which impacted fitness, but because of the onset being later, that they were able to pass it on to the next generation, enough of them to then make it to, ultimately populate Denmark in the 1700s, where we know for sure they were, and then coming across the Mormon Trail to Utah.
There is a question in the chat, which you may be able to access yourself, but I'm also going to read it. Fascinating work. Thank you. Please speculate about gene-environmental interactions for people with these genetic risks. Might there be some environmental risk factors, such as adverse events like experiencing violence or racism, that might influence expression? Looking forward, what are implications of genetic therapy to lower genetic risks or expression?
Yeah, yep. So in the context of the risk alleles for atrial fibrillation, these have low impact. So these are going to be anywhere between increasing the risk, say 1.5 to a maximum of 2.5 times. So what that tells you is that the vast majority of carriers of that risk allele, of the common risk alleles, are not going to get atrial fibrillation actually. And this is where the environmental interactions are so crucial, because that can help drive you into the space where you start to get disease manifestation. And so when you think about, the disparities that we have in this country, these are healthcare disparities, right? That influence who gets treated and with what medications, and who comes to medical attention and who gets the best treatments, et cetera, et cetera. And so those disparities play a major role in our health outcomes, and they dominate the genetic influences. That's for things like common variants that we find in these GWAS studies. When you look at the more rare, high impact, there's no question that environment can play a role as well, but they tend to be more penetrant, to the point that irrespective of your socioeconomic status, you can manifest the disease. And that's why we see lung QT syndrome across all different strata of socioeconomics. Now, survival varies again with socioeconomic status, and so there isn't really a good way we can get around these health disparity impacts on health without directly correcting them, which is something which is very difficult to do, especially in this current era, this current political era.
I've got a question, Marty. So I have an interest in Turner syndrome, and in Turner syndrome, it's interesting because it kind of phenocopies the male risk for a lot of cardiovascular disease. So there must be something about the second sex chromosome that is protective in disomy X females, whatever. But so it raises the question in my mind: have you looked at sexual dimorphism in risk for atrial fibrillation, relating to, as you go through history? Have you looked to see if there's a male female difference in risk, and then what might be causing that?
Yeah. No, we haven't done that experiment. My experience with the risks in congenital heart disease are related to males that have a higher risk for things like bicuspid aortic valve and coarctation and things like that. And, the potential explanation for that is that, men are missing a portion of the chromosome, which would make it an X chromosome. And if you're missing that region, then you only have one other allele, which is the other X. And if you carry, and that's basically the X-linked disease processes. If you've got a genetic variant or a mutation that's on the X, it's going to be more impactful in males because males don't have the corresponding region, whereas females do. And so females would be carriers and males are affected, like, for example, hemophilia. But there's definitely examples in congenital heart disease as well, where there are some variants localized to the X chromosome that function in kind of an X-linked effect.
Further questions on this? Oh yeah, there's one more.
Marty, I'm interested in hypertrophic cardiomyopathy, and as you know, there's not just variable penetrance, but also variable expression. In the same family, some patients present with more of a heart failure phenotype, some patients present more with a lethal arrhythmic phenotype, some patients present with atrial arrhythmias and atrial fibrillation. Are those who expressing more of atrial fibrillation, is it more of one of these genetic influences from an ancestry that's coming where they have both those influences and not just, we tend to think of this very mechanistically. Yeah, we think, oh, yeah, their left ventricular end diastolic pressure is elevated, they have diastolic dysfunction, so they get left atrial enlargement, blah, 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 and those who have more enlargement are more susceptible to atrial arrhythmias. But is that a bad way of thinking, or is it just too complicated?
Yeah, it is. Yeah, it's a big unknown. And if you take, again, you could say the same thing for cardiomyopathy. You can say the same thing in long QT syndrome, right? Like we've got families where there are completely normal QT intervals, and other patient in the sibling has, a QTC greater than 500 and an ICD and everything. And it's like, well, how is that possible? It's possible because you share 50% of your DNA with your sibling, but the 50% that is different, that's a big amount of genetic information that can modify the expressivity of the mutation, even in a monogenic disorder like long QT syndrome, which is highly penetrant. And so these genes that modify the presentation are really not known. And as hard as people have looked, one person was funded to use iPS cells to sort out differences between one patient, one sibling and another. And ultimately that work got published, but it just wasn't, it just didn't really answer that question at a scale that we could really understand why patients present with a particular phenotype and why they don't. And they're in the same family, and so presumably the environment is similar. So I think there's just so much that we don't know about the genome, and so much that we don't know about the non-coding portions of the genome, where the enhancers are, which can influence gene expression as well. And so maybe in the next 10 years, that will get sorted out, and we'd be like, oh, simple explanation. Now we all understand it. But for right now, it's just a big black box.
Maybe one more comment. In pediatric cardiology and medicine, really, I think many of us are aware of a few founder populations in North America. One of them being the one you studied, the other one, Quebec, where a population of close to 6 million is said to mostly stem from somewhere around 6,000 people in northern France. So unique study populations. Unfortunately, we are at the end of not only this lecture, but also our day with Dr. Tristani-Firouzi. It's been a blast. It's been really enlightening, stimulating. Thank you so much for spending the day with us virtually, and hopefully someday we can do the in-person version of this.
Yes, I again, my sincerest apologies for not being there. There's nothing beats in-person contact and face-to-face discussions and collaborations and things like that. And that wasn't possible here, but it was great to be able to see the fellows this morning and talk to them, be at that case presentation, and then just the discussions that we had now and at right after the grand round. So anyways, I really appreciate Dr. Silverbach, your invitation, Lars, your invitation as well. And I'm honored to have been considered for this. So thank you very much.
Thank you very much for spending the day.
My pleasure.